You know, it's like,
you said it was so much different though.
it is so much different, but like they covered it, like the same thing, but like the structure is so different that it looks really different. But like amazing, like orange colored background, like it was like all the reactions. And I compare it. Yeah, this is almond.
It's almond. Okay. Like if you look in the reaction, reaction database, I'm just gonna print it out and put it on the sheet. So like, yeah. There's some that are like, I thought it was all stuff that like we weren't talking about in class, but like those ones are on there too.
You just didn't refer to it like you usually do.
Yeah. So I was like, I was like, I don't know. But like one of them on the practice worksheet was like HBR was adding in. I think it was to an oxide. So I was like, oh, like it'll. I know, and I was like, this will happen. It'll break the epoxide, whatever. But then I was like, he has it as like I don't know how you do it. Do you take the hydrogen off HBR first, then add bromine in, and then the hydrogen and oxygen, make it alcohol. Because I think you just add in to start, then all of HBR would add in. You can't add them. So I was like, I don't understand. And he's like, acidic conditions of HBR is like acidic.
HBR is acidic.
So it's like very acidic conditions. So they add the most substitute from the epoxide. So it was like this. So like adds here. So BR would be there. The epoxide would break. The oxygen there would make sense. So like how do you add it in before bromate is added? And he doesn't give like the steps or anything. They're just like
acidic condition.
And I'm like,
Yeah.
Or about the hydrogen.
yeah, just solution.
So,
Yeah, but I was like, so if you did, when you used to do it, you had the hydrogen off of HBR, whatever it is, usually it's protonate, then you have BR, then it adds in. But you can't protonate it until BR adds in because we talked about it break.
or possible, but no, I don't think it's but like is it possible to extract this for like hydrogen in the solution so that they
It's just H2 and then BR goes in and then you add one.
Right.
The pK matter because like the surrounding acidity is higher than like HBr, H is easier to come. Worse than like yes.
Can you go to the whole thing?
I am going right now.
Do you understand like the multiple choice? It's so deep.
I felt it in the class. How do you really say that?
Yeah, that's what I'm wondering about that.
But yeah, I mean, you know, at least we got answers.
I'm hoping to just break it.
I know.
Give you a lot of pressure.
That's my goal. This one's gonna be harder than the
Yeah.
last one.
Last one.
Our goal was to get an A for both exams.
Orgo?
But we don't have to take that final, but I messed up. Session 1, 2, 1, 2, hold out everything. Because I saw that last exam, last year.
Just make sure I just took my photo.
Yeah.
Like, I don't know, the third exam from last semester. I feel like it's not the final, but still, it's like, it's itself is. Essay exams.
What did you get over?
I got it. Like, wait, no. Exam-wise, it was like A-. So it's just like, I'm at that threshold, like, having to take or not. Yeah.
It's just do your best.
All right. Good morning. How are you guys doing today? Enjoying a little bit more sunshine today. It's gonna get up to 70, I think. That'll be nice. All right. So I don't know if you guys saw, but I did put in the exams for the people that took it. I can't pass it back until everybody takes it. So I will pass it back on Monday. But the average was around 67% this time. And I did curve it 6%. So the grades that are in was an additional 6%. And so, yeah, everybody still is doing really well in the class regardless. Like I've said in lab to some of you guys, that the labs are all 25 points each, and so each time you come to lab and you're participating, or if you're doing the makeup assignment, if you can't be there and you let me know in advance, then you're getting 25 points. Those add up, and so that's bumping everybody's grades up, so there aren't any exams on those, and also the worksheets aren't graded for, it's the answers are correct or incorrect, they're graded for participation as well. So, yeah, if you guys just keep doing what you're doing and studying, you should be fine. Any questions on anything like that, administrative matters? All right. So today we are going to get into Unit 3, and this is actually probably one of my favorite sections to teach and just to learn about. We're going to be talking about microbial ecology. And so, bacteria, they're constantly going into different environments, and basically they have to alter their physiology and their profile expression. And so today we're going to kind of zoom in a little bit on, how bacteria interact in the environments that they're in with each other and with the host. So today we're going to be talking about content in chapter 21. It's a longer chapter, so we're just going to be focusing in on section 3 today, talking about symbiosis. So, yeah, like I said, today we're going to be talking about ecology, or relationships in microbial communities and in connection to humans. So humans, we are an ecosystem. Let me grab my pointer. Okay. So humans, we are our own ecosystem, and basically we are an ecosystem within other ecosystems. So our body is made up of several compartments of very different environments for things like microbes to grow. And so we have here the upper airway microbiome, that's its own ecosystem, and then we have the lower airway microbiome, which is separate from the upper airway microbiome. Different microbes can grow in different parts of the airway, and the same goes for the gut microbiome. We have our skin microbiome, we have just so many different microbiomes, and so all of this then is its own ecosystem and exists within this whole world that we live in that is its own ecosystem as well. So that's the built-in environment. The built-in environment consists of
things that, us, the host, can modify it. So host modifying behaviors are diet. That's one of the #1 things that can influence the microbial populations in our body. Smoking, taking medications like antibiotics that we've talked about multiple times, and that will kill bacteria and disrupt those interactions. Stress is a big deal. We talk about stress as this thing that isn't physical to the touch necessarily, but it 100% can affect your physiology and your microbiome and your health. And so those are all host modifying behaviors. There are certain allergens that can influence the ecosystem within us. People can be allergic to mice, dust mites, cockroaches, pollen, egg white, eggs, egg white protein, which is called ovalbumin, and then fungi. Fungi here is listed under allergens, which is actually the person who actually made this figure is Ari Kozik. She's a professor at Michigan that I actually work really closely with, and so her, this paper that she had basically talks about ecological interactions in asthma and how that can affect your microbiota and your immune responses. But this is actually interesting because it's been shown that having fungi like Candida albicans within your microbiome and having an overgrowth of it can induce a response that's similar to an allergic response. And so that's why that's listed under here. You don't have to necessarily know the details of that, but just an FYI. And there are also, host modifying conditions. So these aren't behaviors, these are conditions. So if you have, a condition already, like an inflammatory disorder, an autoimmune disorder, chronic infection, bronchitis and things like that. So I'm just kind of giving you a little overview of all of the ecosystems that exist within us and the world that we live in as its own ecosystem. And so throughout this course, I hope to introduce you not only to these important concepts, but also important people in the field, because this is a hugely emerging field right now in research, and this is actually right exactly where I fit into all of this in my research and my dissertation. So just a little here.
I'm Dr. Jack Gilbert, a professor of surgery at University of Chicago and group leader for microbial ecology at Argonne National Laboratory. It's an exciting time to be a microbial ecologist. As a microbial ecologist, I get to work with bacteria, fungi, viruses in every environment on Earth. I'm actually having the opportunity to explore this in oceans, soils, plants, even hospitals and patients. It's a telltale sign that I'm a marine microbial ecologist and I'm now a professor in the Department of Surgery. This is because the medical community, the clinicians, are starting to take very seriously the problem of ecology and ecosystem dynamics in a patient. We need to look at the body as an ecosystem, and by doing that, we can develop novel ways to make our patients better, keep them out of the hospital faster, and also enable them to recover to a better state of health more rapidly.
Below, but it's going to be up in the slides if you guys are interested in that. But yeah, so the point here is that microbes, they don't exist necessarily in a vacuum. They rarely live as single free-floating organisms, and so they have to form these populations and communities. So I'm going to go through some key terms that you're going to be seeing. So microbial ecology is the study of microbes in their environment and their interactions with each other. So it's that ecological, community and the interactions of all the microbes. And so microbiota and microbiome usually, sometimes can be used interchangeably, but there are slight differences there. Microbiota refers to the microorganisms of a particular site, an environmental habitat, or a geological period. So basically, if you have, your gut microbiota, that's basically what we're talking about, is all of the microorganisms in that site. And microorganisms, remember, can be bacteria, fungi, viruses, all of those in a particular site. That's its microbiota. The microbiome, though, is the collection of the microbiota, these microorganisms, and their genes, and inhabiting that particular environment. So The microbiome actually includes the genes as well as the microbiota. Metagenome is basically the genome of the whole biological communities from a particular habitat. So meta, the whole genome. And so with metagenome, basically an example of that is the human metagenome. So our metagenome, it consists of all our microbes, but also all of our cells and all of our genes. So it's the whole biological community. And then the metatranscriptome, that comes from the word, transcriptomics or transcription. And so basically in transcription, we're making RNA from DNA. And so the metatranscriptome is the total content of gene transcripts, or the RNA copies of the gene in a community. So just some terms that you should be familiar with. So hopefully you've seen this classification scale before, but just to kind of orient you a little bit, everything has this classification scale, including bacteria as well as humans or eukarya, and it goes from domain, phylum, class, order, family, genus, species. And so basically here we're looking at the domain, bacteria, they're prokaryotes, but for us humans, we are eukaryotes, so that would be our domain. And then as you go down, to the phylum class, we're chordata, mammals, we're primates, and then hominidae, homo, and then homo sapiens. So basically, you don't have to memorize any of these, but kind of just understanding where these terms are coming from. And so for bacteria here, we're talking about a lactobacillus species, Eldebrukii. And so basically, we go down this classification scale. And so a lot of analyses, for instance, will stop at the family level. And then sometimes, you'd want to know more about, what kind of species it is. So you'd want to go down to the genus and species level. So you get more specific as you go down. And so just a little bit more context of terminology for you. So typically with bacteria, the strain level is the smallest and most defined level that we can get down to. But usually, sometimes bacterial strain is used interchangeably with bacterial isolate, but they're not exactly the same thing. So you can have two different strains of the same species. So remember, we have species here, and then there could be strains beneath that. And so two different strains of that same species are also different isolates. And if you have two different isolates that are the same species, they could be either the same strain or a different strain. This might get a little confusing, but just kind of bear with me here. And basically, if you have two different strains, but they come from the same species, they can differ by as little as one nucleotide, and they are now completely different strains. So isolates are an isolate, something you're isolating on a plate. And so if I isolate something on a plate, so this, for instance, is an isolate, this is an isolate, they can either be the same strain or they could be different strains. But if I have two strains of something, they're going to be different isolates. Any questions so far? Okay, so we've talked, been throwing around a lot of, these terms on microbial ecology. Well, what's an ecosystem? It's a dynamic and interconnected community of living organisms and their non-living or abiotic surroundings. So an ecosystem consists of everything that's living and then all of the abiotic factors that can influence those living things as well. Here's a nice picture of an ecosystem. And so the key part is that it includes those non-living surroundings. And so those non-living surroundings can be temperature, humidity, pressure, pH. And so this classroom is an ecosystem of its own. And so it occurs, throughout all facets of this planet and all these different environments. So an ecosystem basically considers the types of microbial activities that are possible. And so those activities of those microbes in that ecosystem are a function of the features of the microorganisms in each habitat. So what that means is the types of different microbes that can be there, so the species present is inside of an ecosystem. That also includes how big the population is or how small it is, and then the actual physiological state. So are they under attack? Is there an environmental stressor that's present in that ecosystem? And then it also includes the rate of microbial activities in an ecosystem, and that's controlled by the nutrients and the growth rates and those conditions that prevail in that environment. So with types, we have basically which organisms are actually there, their population size, is there an environmental stressor present. But then in an ecosystem, we also have to consider the rate. So who is doing what and for how long and when, and all of that is controlled by the surrounding and what nutrients are available. And so the abiotic features can impact the biotic features. And so every species has an optimal environment in which it performs particularly well. So the combination of abiotic factors in that environment, in that ecosystem, it fundamentally determines whether a species can persist there. And so, like I said, all species have limits to the abiotic or non-living conditions that they can either tolerate or not tolerate. So some of those features are, extreme temperatures, humidity, salinity, pH, and we call that the range of tolerance. How tolerant those microbes can be to those abiotic factors. And so the range of environmental conditions under which an organism can survive, metabolize, and grow or reproduce is called its fundamental niche. So this is a term you're going to be seeing. And there are many different interrelationships between microorganisms that can occur between themselves, their living environment, and their non-living or abiotic environment. And these interactions are dynamic, which just means that they can change based on stimuli. So range of tolerance and fundamental niche. So the fundamental niche is where a certain organism or a species can do all three. It can survive, grow, and reproduce. That's the fundamental niche. And so you do this and consider any property, not just temperature. So the same principle can be applied to any factor. It can be pressure, salinity, pH, anything. And so you can see here that it can survive at really low temperatures, for instance. This is how well it can perform or survive. As you get higher in temperature, it can survive and grow, but only when it gets to an optimal temperature is when it can do all three, and so that would be its fundamental niche. Doesn't mean that it necessarily doesn't grow at any other in any other context or niche, but that's its fundamental niche. And so for every microbe, there exists at least one niche where it will be successful. Otherwise, we won't have that microbe anymore. And so I want to introduce another term called a realized niche. A realized niche basically is also considering that interaction with other living factors in that area and where it can be most successful. So the microbe is most successful in the realized niche, but it can also inhabit other niches. And so in other niches, it's probably less ecologically successful than in its realized niche, but it still may be able to compete, metabolize, and reproduce. And so a fundamental niche, ideally, would be, basically you out in the environment and being able to live and survive and do your thing, but my realized niche is curled up in a blanket in bed, and I'm totally fine also super happy, comfortable, and able to survive. I just love that. Okay. So if the fundamental niche is the range of environmental conditions under which an organism can survive, metabolize, and reproduce, then the realized niche is a subset of that fundamental niche. Any questions there so far? So now just kind of going through a couple more terms. When we talk about populations, we're talking about basically a cluster of cells that is derived from a single cell or a single clone of a cell. So all of these cells you can see are purple, so they all come from the same genetic strain. And these populations can grow up as colonies, which just means that they're aggregates of few dozens to few thousand cells. So when you see a colony on a plate, that's basically a few dozen to a few thousand cells, and so that's a population of the same genetic clone of a cell. And populations then interact to form communities, and so a community is a unified assemblage of multiple populations that coexist and interact at a given location or a habitat. So the combination of community and the habitat and the population can form a functional self-supporting system, and that's what we call the ecosystem. So the study of microbial communities is actually relatively new, beginning with the germ theory of disease in the 1900s and even into the 2000s. Basically, there was this perception that stemmed from the focus of most microbiology research being concentrated on organisms that we could grow in the lab as cellular monocultures. So, we basically were so centered in on microbiology in the context of disease and pathogens, and we would take organisms and try to grow them up in a lab, and usually grow them up by themselves, in a flask under ambient conditions. And so this is a very pathogen-centric approach, but that's not completely right. And we know that human pathogens, they account for much less than 1% of the total number of microbial species on the planet. And so that's a good thing, because if it's 1% of all the microbes on the planet that's causing damage, then I'm glad it's not more than 1%, right? But for a long time, we were focusing on these as, when talking about microbes, and we think that microbes are bad for you and they cause disease, but we actually, have to study microbial communities as a whole and their interactions and their reassembly dynamics in order to really understand, how they are influencing that ecosystem. So the vast majority of microbes, including human-associated microbes, they exist in these interactive, dynamic communities. They don't necessarily exist on their own like that in the environment. And that's kind of what makes it difficult when doing lab research with microbes, is it's hard to really replicate that entire system, that whole environment, when you're trying to study them, because they really depend on all of those factors, including other living members, other microbes around them, as well as the abiotic factors in that community. So today we're going to talk about symbiosis in terms of ecology. And so symbiosis is these close, prolonged physical or metabolic interactions between two or more populations. And symbiosis doesn't necessarily mean that they can only, mutualistically live happily together. It can also mean that they live together, but there is either commensalism, where, one organism is benefiting, the other doesn't really care, or it could be a parasite. Those are all symbions, because they can exist together in a prolonged physical and metabolic interaction. And so within a biological community, there are many different types of interactions that occur between populations. We're going to focus on, basically the interactions between microbes and other microbes, as well as microbes and humans. But there are also, I really want to stress that there is microbial and plant population interactions, there's interactions between microbes and animals, and then microbes and insects as well. And so microbes are just really, really important for interactions with everything, every living thing. And we can characterize the type of relationship that populations have with each other in a few different ways. And so there are mutualistic reactions, synergistic, commensalism, amensalism, parasitism. Neutralism is also another one that is not on here. So this is kind of a summary table of these interactions, but I will go through them one by one for the rest of this lecture. Any questions at this point? Can you guys hear me okay with the air coming on? Okay. So, mutualism is a two-way relationship between populations in which both populations benefit. So, both partner species benefit, and they may fail to grow independently. So, they really depend on each other. And they're codependent, as you can see here. That's just kind of another way to remember mutualism. So the main really key example is lichens. So we talked about lichens in probably lecture 1 or 2, but just to bring this back, a lichen is an association between an algae and a fungi, and basically they have to come together in order to create this lichen. And so they cannot exist on their own, but they need each other in order to be a lichen and have all of the positive benefits that lichens do. Synergism is similar in that it also, occurs between populations in which both populations are benefiting, but they can live independently as well. So in the lichen example, they can't live independently, but you can, in synergism. And so a good example of this, also what I have talked about previously, is this mycorrhizal relationship, which I'll kind of refresh your memory a little bit on the next slide. And so basically it is a boost or a bonus if they have each other, but they can live on their own as well. So an example of synergism is this mycorrhizae, and as I said before, myco means fungus, rhiza means root, so it's a fungal root, and it's a symbiotic association between the plant roots and a fungi. And so basically the plants are dependable in an abundant source of carbohydrates for the fungus because of photosynthesis. And so the fungus benefits the plant by improving its access to water and nutrients, because the hyphae, remember, are these long filamentous structures that can extend the volume throughout the soil for the plant. And then, like I said, the fungus benefits from the plant because the plant can undergo photosynthesis, the fungus can't, and so it's a source of carbohydrates. They can still exist on their own, but they do better when they are together. So it's not a codependent relationship. So there's obviously benefits to having these positive interactions between biological populations where both organisms are benefiting. And what that means is you're enhancing the survival capacity of that interaction or interacting population together. And so basically, you're permitting more efficient use of available resources than can be accomplished by an individual population growing alone. And sometimes populations coexist in habitats where neither could exist alone, and that was called mutualism. And then the benefit and the cost may not necessarily be equal across the relationship, but there's got to be a benefit to both. So to give you an example of bacterial mutualism, so this is a growth curve, and And on the x-axis here, we have time in hours. On the y-axis, it's just an arbitrary growth unit. And so here we're looking at two different microbes that exist, and those exist in your gut actually. So Lactobacillus aeruginosus and Enterococcus faecalis. And so you can see here Lactobacillus aeruginosus in the blue on its own, it doesn't really grow well, and same as Enterococcus faecalis. But if you put them together in a flask and you measure their growth rate, you can see that at about 30 hours, they are really skyrocketing. Their growth is really good. So so on their own, their growth units are substantially reduced for both of these species together. But when they're growing together in cultured medium, the growth curve is exceptionally higher than it is for either one of them alone. Can anybody guess why this would be? Like, what could they be doing for each other in order to, you don't have to know the specific details of what they do, but like, what is an example of something that they could provide for each other in order to be able to actually both grow? Like, why can't they live alone? Why are they codependent?
Kind of take a stretch at it, but depending on the environment that they're in, like if it's more acidic or more basic, or the other microbes around them, they could use horizontal, transfer the DNA back and forth.
That's a really good thought. I didn't even think about that, and I'm definitely sure that happens in microbial communities for sure. No, that's a good example. I never thought about that one.
I just read about it when reading over my research.
Yeah, that's more complex than what they actually do, but that obviously does happen between organisms. Basically, what they do, though, is they cross-feed essential growth factors. So one of them produces something that the other one needs to grow. They basically can feed each other. They provide some sort of resource. So these essential growth factors, they're essential for them to grow. L-arabinosis requires phenylalanine, which is amino acid for growth, and that's provided by Enterococcus faecalis, and Enterococcus faecalis requires folic acid for its growth, which is provided by Lactobacillus aeruginosus. So in minimal culture media, they can grow well together, but neither population can grow alone. So they basically provide a food source for each other. So there can also be these one-way relationships, and these can occur basically between populations in which one population benefits and the other is unaffected. That's commensalism. And so this is common, and it often occurs when the unaffected population is chemically or physically modifying the habitat in a way that the second population benefits, and it's a part of its modification process, so it's just existing and it's modifying its own environment through its own function. It doesn't get affected itself, but it does affect another population in the way that it will benefit. So an example of that is in our human GI tract, in the gastrointestinal tract, we have anaerobes. Anaerobes are microbes that do not like oxygen. They are anaerobic or anoxygenic, so they grow without the presence of oxygen. And there are two types of anaerobes. There are facultative anaerobes and there are obligate anaerobes. Obligate anaerobes means that they are obligatory anaerobes, so they cannot grow in the presence of oxygen. Facultative anaerobes are anaerobes that don't necessarily like oxygen, but they can use up a little bit of it, and they can still grow in the presence of oxygen. And so this system of having both in our GI tract is really like such a smart way that we've evolved, in that in order, we still depend on obligate anaerobes in so many ways. They are so good for short-chain fatty acid production in our gut and producing, positive health benefits for us. But, oxygen is in and around us everywhere. And so, how do these still survive till this day after billions of years of having microbes on the earth? And it's because we have these facultative anaerobes that remove the molecular oxygen in our gut, and that allows these obligate anaerobes to be able to persist and live in the human GI tract and do their thing for us. So they're always interacting with other microbes. They're always doing something to help each other. So facultative anaerobes in this interaction remove the molecular oxygen because they basically chemically or physically modify the habitat based on their own metabolism, and that allows the obligate anaerobes to benefit from this interaction. So it's a commensal interaction. Does that make sense? And so another example of this is, production of a biofilm, a bacterial biofilm on the surface of a ship. Basically can then, it's just, this biofilm is being produced, it's colonizing, it's just a community of cells, it's just what it does in that environment, it's modifying it physically, and then that permits a secondary colonization by other microbes. So that, allows other microbes to kind of latch on as well and benefit from it just being there as a biofilm. And then primary infections by one microbial species. So if you have an infection by one species, it may allow other opportunistic species. So they are opportunistic, they grow under certain opportunities. And so to establish secondary infections. So this is a commensal relationship that harms us, and that first one is a commensal relationship that will benefit us. So it can go both ways, but the theme here and the main point is that one population is benefiting, whereas the other is just unaffected. It's just doing its own thing. So we talked about two-way relationships where both benefit, this one-way relationship where only one benefits, but then there are other ways that both can, either that one or more of these organisms don't benefit. So they're affected negatively. So antagonism occurs when one population produces a substance that is inhibitory to another population. So it's inhibiting the growth of another population, but that other population, that one of them is gaining a competitive advantage to it. So this one is two ways, but one is benefiting and one is being inhibited. And so an example is bacteriocin production by bacteria. Bacteriocin is essentially a type of antibiotic that bacteria can produce for themselves. So it keeps, bacteria can form their own antibiotics in a way, and they, keep other bacteria out, but they are benefiting. And then lactic acid production by lactic acid bacteria during food fermentation. So basically, that helps keep it from being spoiled, like the food from being spoiled by other bacteria. So lactic acid bacteria can, produce this lactic acid that can coat certain food during fermentation, and when it does that, it's not allowing other bacteria to colonize. And so production of other chemicals by microbial populations. So basically certain organisms can produce fatty acids, alcohols, H2S gas, and things like that, that allow it to grow and reproduce, but it inhibits another population that can't live on these things. And there's also some discussion about whether our immune systems and our microbes have this kind of relationship as well, because there's this really complex system going on. This is a little bit of where my research kind of also dips into trying to understand the bacterial reassembly dynamics and how that influences our immune system. And so we'll get to this in more lectures, but just to give you a little bit of foreshadowing there. And so this is, antagonism is essentially this antagonistic relationship, and one is benefiting by having a competitive advantage, and the other is being inhibited. So that's amensalism or amensalism. Any questions at this point? All right, so we talked about the impact of positive interactions between populations, but there's an impact, for these negative interactions between populations. And so basically, this is a way for the ecosystem as a whole to protect itself against unwanted or intrusive organisms. So one way that, an ecosystem can promote this negative interaction is basically it can eliminate a population that's not well adapted for it to continue to exist within the community of that habitat. And so they basically tend to prevent invasion of an established community. So basically, there's this term called colonization resistance, which just means that there are already microbes there that are protecting that barrier, and it's resisting colonization by other foreign populations. So, for instance, if you inhale an allergen, we have microbes in that community and our airways that are protecting that epithelial barrier from the allergen to be able to invade through and cause an allergic reaction. So they are resisting. That's colonization resistance, and so it prevents the invasion of indigenous population or of foreign populations by having those indigenous populations there. So these negative interactions are important because they maintain community stability in the ecosystem, and they kind of act as this feedback mechanism to limit population densities. So it's good to have negative interactions between populations. And it all really comes down to the resources that are present. And so usually this influx of resources is happening in cycles. Like when you eat, for instance, you're not eating 24 hours a day, you're giving your body resources in these pulses or in these cycles. So a large part, resources typically enter an ecosystem intermittently, and this usually is a large pulse of nutrients that can be followed by a period of deprivation of that nutrients. And so microorganisms, like I said before, often face this feast or famine existence. So they produce intracellular storage reservoirs when resources are abundant, and they can then draw upon these reserves in periods during cellular starvation. And so extended periods of exponential microbial growth in nature are likely rare. It's rare to see microbes just growing and growing and growing and growing and always having all the nutrients they need. Usually it's more intermittent. And they typically grow in spurts, and that's linked very, very closely to the availability and nature of the resources. So one thing that has to come into play always is E. coli, for instance. We have this E. coli that we study in labs. We have non-pathogenic strains of it, and in our gut, it takes about 12 hours to double. But in a lab, we can let it double in about 20 minutes. And that's just because we're literally putting it in some culture media, and it's able to grow really fast. So we know exactly what's in there. We know what it needs to grow. In the gut, though, E. coli is not alone. There's other things around it. And, it has the means to double, but it takes about 12 hours because there's other factors. There's living and non-living things in there that are also doing their own thing that's going to affect its growth. And this actually poses an issue in a lot of research studies, because when you're trying to, do these studies that can, hopefully have a potential impact on human health and disease, it's really hard to replicate things in a laboratory setting and replicate what it would be inside of the gut. So you really have to control for things and consider all of those things. And so that's what makes my research very hard. So that's just one example. But you have to really consider that it all comes down to the resources available. And so mirroring on, this negative impact, there's also competition that can occur when two populations are striving for the same resource. So usually often it's for a single nutrient present in limiting concentration. So there isn't too much of this. There's just a limited amount of this resource, and there's two microbes that are competing for it. And so as a result, both populations achieve lower densities than they would be achieved by just one population being there in the absence of competition. So there's no, they can both still grow slowly, and these competitive interactions tend to result in exclusion, where one of them will be able to overgrow or outgrow the other population. And so, but that's not necessarily like, it's not like it's one is benefiting and inhibiting the other. It's not inhibitory, it's a competition. And usually in a competition, there's natural selection. So the stronger one is going to survive. So to give you an example of competition, basically this can lead to exclusion of the less successful population. So here we have these two types of paramecium, P. aurelia and P. caudatum. And so when they are grown separately, this is what the paramecia looks like. It's just a picture of it. So this is over time and its population density. On its own, this one can grow really well, and so can this one. You can see this one grows even better on its own, which is why when you put them together, you have the blue one, P. aurelia, growing really, really well. And then eventually, once this uses up all the nutrients that they both need, this one, P. caudatum, is completely excluded over time when they're grown together in that mixed culture. Any questions here? All right. So competitive exclusion can prevent two populations from occupying the same ecological niche. That's because they both can't play the same functional role at the same location at the same time. So when two populations are competing for the same limiting resource, one will succeed and the other one will fail. And so as a rule, the population with a higher growth rate under the given set of conditions in the habitat will prevail over the population with the lower growth rate. So over here, we saw this one had a higher growth rate on its own. So if I were to give you an exam question showing you these and asking you, telling you that they will compete for the same resources, which one will outcompete the other, you would know that it's the blue one, right? Okay. And so this competitive exclusion is also known as colonization resistance, which is what I introduced earlier. And this can be a good thing. So I'm going to talk a little bit about this term, colonization resistance. And this is essentially what I was trying to explain a little bit earlier. Here I have a nice picture for you. So the gut microbiome prevents the invasion of new species or pathogenic species through colonization resistance. This is exactly what my whole dissertation is centered on. So over here, what I'm showing you is the epithelium. This is the epithelial layer. And so these are intestinal epithelial cells, and these provide a barrier for, things in the outside community to not enter. This is where you have the lamina propria, which is your gut intestinal lining. And so this is a barrier. It's a physical barrier, but there's also a mucus layer right on top of it that coats the intestinal epithelial cells, but also protects as that also acts as a way to protect this barrier, your intestines, essentially. And so we also have, this is called the lumen, which is right outside of this epithelial barrier and the mucus layer. And we have a lot of different microbes here, and these are good microbes. These are what are already sitting there, and they are protecting from things. They kind of competitively exclude, so competitive exclusion or colonization resistance. So they resist colonization of other things in the environment that can be harmful. And they do this through many ways. They're not just sitting there and playing defense and like boxing out anything that wants to come in. They actually have different ways. So they can cause adhesion exclusion of pathogens. Pathogens here are shown in red. And so basically commensal E. coli, for instance, or you don't even have to know these species, but just commensal bacteria basically can provide a physical barrier themselves so that if something wants to come in and adheres to this layer, they're already there. They can't, they don't allow something else to come in. They also limit carbon sources and nutrient sources so that pathogens that will thrive on certain things, they can't, because these are limiting those certain things. They also secrete antimicrobials or antimicrobial peptides that can combat pathogens. And they can also, if they see pathogens, they could potentially deliver some toxins. They can create toxins that will then block these pathogens. But these pathogens are also smart in their own way, and they can express certain virulence factors and do different things that, can cause them to, break this barrier. So typically in the context of, here you have all these good bacteria for you, there's purples, blues, yellows, just a lot of them, different populations creating a community. And so when you take broad spectrum antibiotics, broad spectrum just means that it, kills both gram-negative and gram-positive bacteria. It has a broad specificity for bacteria. These antibiotics will kill off all or most of these microbes. And so when that happens, then these things then aren't produced and aren't playing defense anymore. And so that's when you can have pathogens come in and break that barrier. And so in the context of my research, pathogens don't necessarily have to be something that doesn't exist there naturally. It can be something that is already there that's opportunistic, remember? So opportunistic pathogens are microbes that are already existing, but under certain conditions, they can overgrow, and when they overgrow, they cause disease. So among these bacteria, there's also fungi that exist in the microbiome. There's viruses too, but the fungi example that I always give is Candida albicans. So that usually exists as a normal part of this microbiome, but when given antibiotics, antibiotics target bacteria only. So that depletes the bacterial populations, but then it allows this niche for the fungus to overgrow, and then you can get candidiasis, which is an overgrowth of fungus, and that causes, a whole slew of other infections. And so just in this example, the gut is the biggest site for Candida albicans to cause dissemination and get into the bloodstream and cause death. And this happens a lot in immunocompromised individuals and people who are always on antibiotics, so it can be deadly. And so, really understanding these interactions, because we prescribe antibiotics all the time, and we need them, we definitely do, to help treat illnesses and infections, but we have to understand that when we do that, we're also exposing that environment to potential pathogens and things that can overgrow and cause harm. So that's my thesis in a nutshell. One part of it. All right. Does that make sense? Any questions? Okay. So low population densities and low metabolic activity permit microorganisms to coexist without competing for the same available resources in the habitat. So before this, we were talking about having different microbes. We have a lot of them, and they all are doing their own metabolic activity. They all have their own functions, and usually that leads to competition because they're all existing in the same place. But if you have fewer organisms, lower populations, and there isn't much going on, not a lot of metabolic activity, then they can coexist without having to compete. And so that's a neutral state. That's called neutralism. So basically, low metabolic rates of activity characterize this resting stage of microbes, so they favor a lack of interaction. They kind of just don't really want to bother the other person. And so basically, by being at very low population densities, they're less likely to come in contact with each other. And then actively growing vegetative cells are less likely to exhibit neutralism towards other microbial populations because they're vegetative. And so one-way relationships can also occur between populations in which one population benefits and the other is harmed. So we talked about one-way relationships already in the context of one population benefiting and the other being excluded. And in this one, excuse me, in parasitism, one is benefiting and the other is actually harmed. So to distinguish this from predation, when I think of predation, I think of a lion that wants to eat. And so this is different. Parasitism is not predation. Parasitism is characterized by a relatively long period of contact, so they're existing together and they're there for a long time, and then one benefits, the other is harmed. And so usually it's not, when a lion wants to eat, it just goes and catches its prey and then it moves on. It's not like that. And then also the parasite is usually smaller than the host, which also is different from predation. And so the parasite normally derives its nutritional requirements from the host cell or the organism, and in the process, the host is damaged. So that's how it works. It's getting its nutritional requirements and it damages the host through that process. And examples of parasitism are infectious diseases. So plant and animal pathogens are examples of parasitism. It's not necessarily just a parasite that can cause, that's also parasitism, but parasitism can exist in the context of bacteria and microbes because, infections cause harm to the host, so that's a parasitic interaction. So the intensity of positive and negative interactions between populations, because remember, there's both happening in this community. Those generally are greatest when you have high population densities and when populations are actively growing. So that's kind of, I guess that's pretty, that would make sense. That's pretty intuitive. So under these conditions, when you have high population densities and they're actively growing and, there's a lot of metabolic activity going on, organisms living together within the community interact and they compete for available resources. But there's obviously going to be fluctuations in those environmental conditions that can lead to shifts in those competitive balances. It's not always going to be a straight arrow. There's going to be, different divergences. And this can result in in population oscillations within that microbial community. But there's this term called spatial separation that allows microbes to escape competitive pressures that allow them to coexist in those competitive populations. So spatial separation is just ways that different communities are separated from each other. And this is relevant in the human microbiome a lot because a lot of our niches where we find microbes are separated. So like I said really early on in this lecture, we have populations of microbes in our airway that are going to be different from our gut, that are going to be different from our skin. And so those are separated spatially, and they're connected. Our mouth is connected to our airway and it's connected to our gut, but they are also still separated. So that's what allows those microbes to survive and continue to fluctuate in those environments and exhibit these different positive and negative interactions that it oscillates. So in most cases, a given species has an effect on a small number of other species, but not on the entire community. And so as a result, the extinction of one species usually doesn't affect the long-term viability of the community or ecosystem. Because all of these different microbial interactions, whether it's parasitism or commensalism or any of them, usually it's certain microbes affecting a single species, because they all do different things, but usually extinction of one species isn't going to affect the long-term viability of the community or ecosystem. And so that's why when you take broad-spectrum antibiotics, you're actually targeting a whole broad range of microorganisms, and that can affect the long-term viability of the system. And so usually, other species in the community can provide that missing physical, chemical, or metabolic element to the community that now extinct species was providing. But however, there's always an exception to every rule, and sometimes the loss of that single species from the community can have a disproportionately large effect on the entire community. And that that species is called a keystone species. So the loss of a keystone species from the community can have a really large disproportionate effect on the entire community. And as it suggests, this is a species that plays a role in its community that's really, really important than its relative abundance might suggest. So basically, even if it exists in low numbers, it's clearly doing, playing a really important role in that entire community, and losing that one species is harmful and other organisms can't pick up that role. And typically the most abundant species or the major, it's usually like the most abundant species or major energy producers, while they are vital to the maintenance of the community, those aren't keystone species because they're really, really abundant. If they're major energy producers, it's going to be, it's going to be there, and it's not really a keystone species. Keystone species really usually exist in really low numbers. And so they may be predators, sources of food, synergistic species, or providers of other essential factors for the community. So they usually exist in low numbers, but they have a really, really good effect on that community. And those that are really abundant and have those really big roles, they're really vital to the maintenance of the community, but those are not keystones. So does that make sense? Any questions? All right, well, we will continue with more microbial ecology now that I've introduced you to a lot of these terms. And then, yeah, we'll see you guys on Monday. Enjoy the sunshine. You want to meet me in the lab room, and we'll take you in there? Which lab? Like the micro lab.
Oh, okay, yeah.
Just because my office is like small and dirty.
There.
And better.
I pretty like that's it. That's,
The average was 90% for the
oh, then she's like,